everyone. Today I'd like to introduce Scott Hansen from the Scripps Research Institute, and today he's going to be teaching us about mechanosensory biology. Scott?
All right, Kim, Keith, thank you for the opportunity to present today or teach. So I'll be talking about mechanosensation and, oh, my keyboard's not working. So mechanosensation is this process by which we sense mechanical force. And we're interested particularly in the molecular determinants of that. And I want you to imagine for a second, you're sitting on your couch, there's birds chirping outside, the sun's coming in, you know, it's a great Southern California day. You get up and you're walking across the room and wham, you hit your toe on the coffee table. No part of you is thinking about the birds chirping outside. You're more likely to express foul language than anything else. Everything goes out the window, right? And it's just simply a mechanical act, right? Mechanical force acting on tissue. And it has a very, very potent effect on our emotions and on everything we're doing. And so how does that happen? And what are the molecular determinants of that? And it's been surprisingly recent that we even really started thinking about this. So in humans, we have these sensory neurons, and they're specialized to sense this force. And they have an axon, a long axon, and dendrites. And the mechanical force is thought to act at these dendrites, and ions pour in sodium and calcium, and they depolarize the cell. And this is all one cell. And once it depolarizes, it gets propagated up this axon and then to the spinal cord. And Keith mentioned this in his introduction, that you can take ultrasound and stimulate the tip of your finger similar to mechanical force. And that's expected. Ultrasound is a mechanical force. So interestingly, well, this could be what would cause this. It could be, you know, it's thought to be ion channels, but it could be anything specialized in these dendrites to sense force. But it can't just be the dendrites. And the way we know this, we've all experienced this. This nerve runs along the elbow right here and up through the arm. And we've also been walking along and then bumped our elbow on the table or something like that. And you get, it's not the exact same response, but you get a very similar response, right? Your hand, now think about this. You hit your elbow, but it's your hand that feels the pain. So it's the same nerve that's in there. It's these nerves that your brain doesn't know what got hit, but it thinks that it's your fingers, when in fact it was somewhere along here on the axon. So this tells us the first piece of information. It cannot be just specialized ion channels in the dendrites. The nerves themselves are something very fundamental about a nerve that must sense force. And the question is, and we know this also from stimulation in the brain through ultrasound as well. And the brain actually has lots of force on it. This is from Itmar, who's also at Stanford. And this is pulsating CSF into the brain, and it's amplified so you can see the parts of the brain that actually move. But there's lots of pressure and shear, and all over our bodies we have pressure and shear. Ions also change concentrations in the brain. I know, Keith, you were very interested in Nedergaard's work about falling asleep, right?
Mm-hmm.
Well, it dumps, when you fall asleep, you dump a bunch of ions, potassium, and that changes the salt concentrations. And we know that when you change salt concentrations, the cells can swell and shrink. So these are kind of all the forces that are going on in our bodies, and our cells sense that. So what are the molecules? Well, people have been thinking about ion channels, and I'm going to talk a lot about ion channels. There's two types, and for now I'll just call them inhibitory mechanoeffectors and excitatory mechanoeffectors. And the inhibitory ones release potassium and chloride, and the excitatory ones receive or allow sodium and calcium to come in. And those channels are, for example, TRAC, which is what I'll talk about today, which is a potassium channel, an inhibitory one, and piezo, which is an excitatory one. But these are not the only mechanosensors. There are enzymes. I'll talk about an enzyme called phospholipase D2. And there are many of these enzymes, and some of them we don't even know if they're directly activated by force, but they're clearly responding. One of them is, eNOS. And then there's receptors. For example, G protein coupled receptors. The HR1 receptor is mechanosensitive. And then there are cell adhesion molecules, integrins. So there's a myriad of things that could be mechanosensors, but most people think about the ion channels because it's presumed that only ion channels are fast enough. And that's really, it's not that people don't know that everything else is mechanosensitive, it's just purely the speed of it. And so some very early work led people to think that it could only be ion channels. But
So
we
Scott,
will discuss that.
when you say speed, do you mean the time that it takes for the actual mechanical input to be translated into a neural signal?
Exactly, the latency, that's a lot better word.
Okay, latency.
The time from the receiving the signal to the time you get the output. So we obviously respond very quickly, right? It doesn't take very long for us to realize we stubbed our toe. So what are the molecular bases? Like, what are the theories out there? And one of the theories is disruption of biological order. So one of the early ones was protein folding. You have proteins that fold up, and then you stretch them out and they unfold. And another one, and this is the one I'll talk a lot about, is ordered lipids. So lipids, and I'll go into a lot more detail later. So right now I'll just give you the concept. You have two compartments in a membrane. Those compartments are separated by some distance, and then either the domains themselves or the compartments themselves or the molecules that are in these compartments respond to mechanical force by mixing. This would kind of be like a glow stick. Do you know how a glow stick works, Keith?
Yeah, like vaguely, just two chemicals that fluoresce when they mix, right? So you give it a little shake.
Yeah, you have a glass barrier, you break the barrier, they mix, right? So Halloween's coming up, right? Like we know that this stuff works. Like if you have a barrier and you break it, you can cause a signal. In that case, it's a light signal. Okay, so this is a much more recent development and a very new way of thinking about mechanosensation. Classically, the way people thought of it were the next two categories, and these are mostly limited to stretch. So, for example, if you have a channel and you stretch the membrane, the hydrophobic part of the channel, if it's rigid, will just poke out of the membrane, and that has an energy barrier. And so the way you can reduce that energy barrier is if you have a radius here, and that radius gets larger. So you have a change in conformation from going tall and then matching the thickness of the membrane. Another one is direct stretch or direct activation. So the membrane is elastic, it's gooey. And if you apply a radial force, an outward force through stretch, then this channel here can increase its radius. And so these are the classical models. And I'm going to show a video that kind of helps you understand how that would work. This is a mechanosensitive channel out of a bacteria from the Peng Yuan lab at Washington University. And you can see this hydrophobic thickness right here gets much, much thinner. And then when it flips on its head, you can see that it also expands out. So this was structural information. This is the pore down the middle. You can see it stretching out. So this is structural information that kind of supports those hypotheses. But these are in bacteria. And this was to keep a bacterium from exploding. And we're interested in ultrasound and how, say, shear and other pressures might cause mechanical activation. So for the rest of the talk, I'll talk about this TREC channel that is a mammalian channel. It's a two-pore domain channel. And it's activated by shear, stretch, and ultrasound. And this is a three-dimensional structure in two states. And the main conformational change is this gating helix moves up. And this, at the tip here, colored in blue, is a lipid binding site. And this lipid is phosphatidic acid. When it binds right here, it activates the channel. So this is a lipid-gated channel. And the enzyme that makes that lipid is the PLD enzyme I talked about. Okay, so I think this is kind of the setup about the ion channels. Before I go on to the mechanism, do you have any questions about the channel?
I have a couple. So Scott, in this image, this blue tail structure here, does that just block the pore and then it moves out of the way?
No, it's actually a mystery. They don't really know how it gates it because they haven't, from the structure, it's not obvious how it gates. Mm-hmm. So in the other channel, it was really obvious from the structures what was happening. But sometimes you solve the structure and you think in the open and closed state and you think you're going to have all the answers. And then there's a mystery still there. And they're like, well, we don't know how this opens.
Right. I have one more question.
This is the gating, you know, mutagenesis and lots of other studies. They know this is the gating region of the channel.
So this next question is a little more vague and you don't have to answer it if you don't want, but in the classical models where the membrane may be flattened and actually tug on both ends of the channel, you know, how do those theories suggest that lipids are bound to the ion channel? Because they would have to be connected so strongly that their pull would also pull the channel apart. Is that the case in your opinion? And feel free to.
Yeah, I think the bigger problem is how do you maintain, if you have tension, you would think that there's still fluid. So even if you could get them to open briefly, you would expect them to close right away. And that you would only have one event where you could open them briefly and then they would just close. Mm-hmm. So this idea that you could pull them open for a long time and then release them and the same amount of time every time they close, you know, there's some things that are a little hard to explain. But from a biophysical standpoint, there's very good data saying that at least the magnitudes of the forces are in the right direction. And it's just vague. We don't actually know if that can happen, but that's what's been proposed. So, and if you look at the structure, it looks like it opens that way. So there's some things backing it up. But as we go along today and we talk about the membrane and we start thinking about the membrane, even that first structure we just saw, we might start thinking about it a little bit differently. And that's my hope that you'll see. So before we go on, go ahead.
Yeah, I was going to say, if it's not clear, I've been working with Scott for a while. I'm very biased towards his ideas, but we can consider all options here.
Yeah. So we'll see if I can bias the rest of you, Tim, as well. Okay. So this channel, we need to look at how it responds to ultrasound. So as I mentioned, TREC is a channel that's directly responsive to ultrasound. And this experiment was also done at Stanford by Kubanek. And what he did is there's a plate here with cells sitting on it. And then he focuses the ultrasound onto those cells that are expressing TREC. And then he has an electrode. And this electrode measures the current. And this is the output from that experiment. On the y-axis is the current that's coming out of this electrode. And then he has a voltage electrode that he can turn the channel on and off with voltage. It's outward rectified. So he's holding it closed or inward current. And then when he flips positive, then he starts getting current. And you can see that it robustly activates. TREC is, there's a lot of current here. There's a lot of current for a channel. And here in blue is the time that the ultrasound was applied. And you can see that as ultrasound is applied, that the amount of current dramatically increases. And right here is where it stops. And this is a very interesting observation, that as soon as you stop the ultrasound, you would expect if it's just stretch alone that it would drop back down, right? But it doesn't. it actually increases for probably 200 milliseconds, and then it very slowly starts to come back down, but stays activated. So this might be the first indication, like, wait a minute, what's going on here? Why, if it's just stretch when you release, why is it actually activating more? And then why does it take so long, the membrane to reform or something? It seems a little weird, right? If it doesn't mean that stretch couldn't be involved, but we'll directly test this. So this suggests that the membrane might be involved in this mixing mechanism. You can see that in that would easily fit that model. So before I describe the membrane, you need to know, or before I describe how the mechanism, you need to know a little bit about membranes. How are membranes organized? And what could mechanical force possibly be doing to the membrane itself? And so membranes are organized into ordered lipid domains, which is shown in orange here. They have long chain saturated lipids, and they're thicker. And they have cholesterol, shown here in purple, that intercalates in here and organizes it. And that's why they're thicker and more rigid. There's one other really, really important concept here. And I think this is very much underappreciated. And that's the fact that the affinity for these domains is dictated by palmitylation. So what's palmitylation? Do you know, Keith?
I
Putting really on the spot here.
Kim, do you know?
No, I don't know.
Okay, well, this is good. So palmitylation is a lipid that's attached to a protein, and it's a saturated lipid that's 16 chains. It's a very particular lipid, and it's bizarre. Usually there's two of them, and if you have these two lipids, you migrate to these domains, and you just have affinity for them, and that's where they stick, and it's pretty reliable. There's many, many proteins that are palmitoylated. So my lab has been, and this has been known for 20 years. My lab has been looking at a different type of domain that's located here in the disordered region, so the fluid part of the membrane, and that's PIP2 domains. And this is another type of lipid, and it has two phosphates, and it's arachidonic acid, arachidonylated, so it has arachidonic acid in its tail. So, okay, to understand the difference between these two, it's like having saturated fat and lard that's solid, and having olive oil that's fluid and running around. They're both lipids, but they're very different. One's fluid, one's not. So this one is much more the olive oil type, the unsaturated. So this is your good fats like olive oil, the salmon that you eat. It has these polyunsaturated lipids in them. So that's what's in this lipid domain. There's another lipid domain I'm not going to talk about, but this is just a point, PIP3. But these three domains are all separate. And this channels can be activated by these lipids. But you can also be targeted to this domain by pH domain, SH2 domain, and PDZ domains. So if you have a domain that binds PIP2, then you actually migrate to PIP2. If you have palmitylation, you migrate to GM1 domains. So this will become very important.
What do you mean when you say you migrate to those areas?
So if they're separate in the membrane and you have affinity for a GM1 domain, well, then you would end up in a GM1 domain. If you have affinity for a PIP2 domain, then you would end up in a PIP2 domain. But a PIP2 domain is not the same place as a GM1 domain. So you're going to be in very different places. It's like going to the membrane versus going to the ER. Like if you're targeted for the ER, you're in one place and they're completely different proteins in those locations, completely different signals. So it's important to know which place you went. The question is, what happens when you have palmitylation and a PIP2 binding site? Now where do you go? Do you go to the GM1 domain or do you go to the PIP2 domain? Well, I'll show a little data on this later, and I'll just tell you the answer right now. PLD starts in a GM1 domain. So its affinity for GM1 domains outweighs its PIP2 binding site. But if you apply mechanical force, it changes the interaction of palmitate with this domain here, and it disrupts the domain, and then PLD comes out and it goes and it binds to PIP2. So this is a membrane-mediated mechanism where mechanical force is simply disrupting one of the compartments, and then PLD leaves and it goes. And when it arrives here, it finds PC. I know this is a little complicated, but it's really important.
So wait, Scott, to go back to Kim's question, when you say migrate, do you mean that it basically frees the domain and that random travel throughout the membrane, you know, may lead it to PIP2?
Exactly.
Is that what migrate is? Okay.
Away. It just translocates laterally in the membrane. Remember, this is one plasma membrane. So it's just translocating over to the other set of molecules that are in the domain. And when it arrives there, it finds PC and it hydrolyzes it and makes PA. So this is how the enzyme's turned on and off. Usually you think about an enzyme getting turned on and off with a conformational change. That's not what's happening. The enzyme's the exact same conformational change. But when it's here, there's no substrate, so it's not active. And then when it's here, it finds its substrate and it gets active, but it needs this PIP2 to pull it over to this other domain. And the important finding that will actually show a little bit of data for this is that the mechanical force disrupts this interaction of GM1 with the palmitate. Okay. And so how do we know this? We've only recently discovered this. And the reason is because the distance between, let me go back, the distance between this domain and this domain is really small. How small is small for a microscope?
I was about to ask.
250 nanometers is what we used to always be able to measure down to using confocal and classical microscopy. But it turns out that these two domains are only 42 nanometers apart. So this is a nerve, and we've labeled PLD and we've labeled PIP2. And in yellow is the co-localization of those two. And you can see there's yellow all over the place. So somebody would say, oh, these two things are in the same location. But that's just because the resolution is only 250 nanometers. If you use what's called super res imaging, which I'm not going to go into detail about because it's very technical, but this got the Nobel Prize, I think, in 2014. It's a new technology. And it's a way to look below, down to even 20 nanometer resolution. And as soon as we looked with super res, these are the same nerves. These are just zooming in on that. We see PLD and PIP2 are not in the same place. They're actually separated by 42 nanometers. Anyway, so it's important to realize that sometimes you're looking at a microscope and you think they're in the same place and you're just totally being fooled by the fact that there's a whole world that's very small that we couldn't see even 5 or 6 years ago.
So Scott, are the colors also mapped correctly? Like the blue in that image is actually GM1 tagged with a fluorescent protein and vice versa?
No, actually in this one.
You flipped the colors.
Yeah. Well, I added this later. Yeah, the blue and the red get flipped around a couple of times. You'll see a couple of times. Yeah.
Those are the two molecules in different colors.
Yeah, the blue, actually, because it's PLD and PIP2 that's actually labeled. That's why.
Okay. Very cool.
Okay, so if we want to see this happen, well, first, I just want to give you kind of a picture of how the membrane's behaving. So this is just labeling the GM1 domains. And so just watch it for a second. You can see things kind of moving around. This is what we assume is a GM1 domain. It's labeled by a GM1 probe. And if you look at it in more detail, you can see that that they also, the nodes, there's little nodes here, and then they exchange proteins along a specific track. And really what I want to show is that this is kind of dynamic. And you know, the lipids aren't just one little raft, basically. Things are exchanging, but they're exchanging in an ordered way. Does that make sense?
Mm-hmm.
Okay, now we want to see it with mechanical force, right? We're interested in mechanical force. How would mechanical force disrupt that? So we have to trap it in a mechanically activated state. We grow the cells that we want to mechanically stimulate in the shear chamber. And then we pump fluid in here and across the cells. And we can, using a pump, very carefully determine exactly how much shear is being experienced by those membranes. And I liked your explanation, Kim, of shear and propagation of waves. It's very much important for this concept. So if those waves, those surface waves then are going through the surface of the cell, then we apply a fixative while the waves are moving, and we can trap it in that fixed state. Is that clear? So now we want to know, okay, what happens? Where does PLD go when we mechanically activate it? And now we have the resolution. We can actually look at it. So this is the super res imaging data. And in this first set of data right here, we've labeled GM1 and we've labeled PLD2. And so when we have no shear, which is in blue, we can see very good correlation. That's what's on the y-axis here. And we want to look at short distances. So at short distances, it's highly correlated. And then as you get further and further away, the correlation starts to go away. That's what we expect, right? Now we apply this shear. And this is 3 dynes of shear, which is substantial, but shear that we experience in our bodies. And you can see that PLD is no longer associated with GM1 domains. It's pretty dramatic. So PLD is no longer here and it's no longer associated. And the question is, where did it go? So we can tell where it went if we label PIP2 and PLD. So that's what's here, PLD and PIP2. Now we should expect the opposite. Before we add shear, there's very little association. And then after we add shear in red, now we see greater association. So this is at a very, very small level, very, very high resolution. We can see that PLD left a GM1 domain and went to the PIP2 domain, and in response to mechanical force. And if we just look at what happened to the domains, they did get smaller. So this is size on the y-axis. They all got smaller, both PIP2, but GM1 domains are a lot more affected. And in the end, GM1 domains are actually smaller than PIP2 domains after the mechanical shear. Okay, well, how would this then lead to activation of a nerve? This is what we want, right? We want to understand in the brain, when we're activating a cell, how could perturbing these membranes then lead to activation of an ion channel? So in the introduction, I mentioned this enzyme that we've been discussing, PLD, binds to the C-terminus of TREC, and it produces the phosphatidic acid. So now the question is the channel really mechanosensitive, or is the enzyme mechanosensitive? Remember, everybody had always thought that the channel was mechanosensitive because they thought it had to be the channel because that's the only thing that would be fast enough. So we're interested in the latency, how long does it take, and is PLD really the mechanosensor? So how would we test that? Well, this is our hypothesis. PLD is coming out, binding, and then activating TREC. So what would happen if we had a catalytically dead PLD? So that's in this experiment right here. If the channel is activated by direct stretch, it shouldn't matter if PLD is there, but should just respond, right? If it needs the enzyme, if the enzyme is coming out and it's the enzyme, then when we put a catalytically dead enzyme, then we can put stretch on it and nothing should happen, right? And it should be very easy to tell which way it is. And so we designed this experiment using electrophysiology. The channel is in a cellular membrane that gets pulled up into a pipette. This is the pipette right here. And then we have negative pressure. We just pull on the membrane, and these membranes pull in further and they stretch as they go into the tip. And you can see here, this is the current. I'm guessing the students don't normally look at electrophysiology curves, right?
I think Steve's lecture is before this one, Kim. So they should have some familiarity, Scott. You can talk about it.
So this is the current on the y-axis, and then this is just showing the amount of pressure we add. So we add more and more force. This is stretch force, but you could think of the same thing as if you add more and more intensity of ultrasound. And this channel, as you get more and more force, opens more and more current. And it stays open for the duration of the stretch with a tiny, tiny little bit of desensitization, but not much. Okay, so what's going to happen now when we change this out and put in a catalytically dead enzyme? And the answer is 0 current, absolutely none. And this was the first indication that this channel is not responding directly to this stretch in the membrane. And remember, this is a classic mechanosensitive channel that everybody thought was responding directly to mechanical force. And so this needs to be explained. Well, if the enzyme is what's mechanosensitive, then that is the mechanosensor. And then it turns to the membrane is the mechanosensor, not the channel. And that's a really important finding. Of course, we need to explain, could this happen really quickly, right? Because then people say, wait a minute, this channel is responding in low millisecond timeframes. How could you have a second messenger cascade that then goes and acts on mechanically sensitive channel? So, oh, sorry, I missed one experiment. To confirm this, we cut off the C terminus so that the enzyme was present and the channel was present, but they just couldn't interact. And you can see here that cutting off the C terminus also had the same effect as mutating the enzyme. So we're pretty sure that it's this interaction that's important with the enzyme and the channel. Okay, so how fast could this happen? This is latency. So theoretically, an enzyme can diffuse from here to here in 650 microseconds. And the important point is that this distance is so short. It's really, really short. And this is very difficult to conceptualize, really small distances. Excuse me. I should have got a drink. So my throat's drying. So what happens if you increase PIP2? Well, this should get shorter and shorter distance, right? And you could even have very, very fast activation if the distance got short enough just by increasing PIP2. Well, we have the ability to test this. And that's because all the current was from PLD. So we can just look at when the current from PLD turned on. And this is our current trace. This is what was coming out of the electrode. And this is the time when it started. And we know that the instrument has a calibrated delay of 6.9 milliseconds. That's just what it takes the instrument to go through its responses and finally arrive and cause pressure. So that at 6.9 seconds, we can see almost immediately the current starts going up. And by the time we get to 2.1 milliseconds, we have statistical significance. In other words, when we reproduce this, this is always statistically significant by 2.1 milliseconds. So we know that this is low millisecond timeframes, and this is as fast as we can measure. So basically, this isn't limited by, this is limited by how fast we can measure. It could be going faster than this. We just can't see anything faster. So to us, it appears instantaneous. And we know that this is the PLD current, right? This is not the channel alone. This is the PLD current, which means that the membrane is capable of responding in a second messenger fashion immediately, as long as you're at very, very small distances. So remember all the mechanosensors we talked about in the beginning. Now everybody needs to consider all of them, because if they can all go at this pace, we don't know, you need to look at very small distances, right? But all of a sudden the ion channels might not be the only thing that is mechanosensitive in the brain. And of course, if you're adding ultrasound, you're going to be activating, you're going to be disrupting all kinds of these very nanoscopic compartments in the brain. Is that clear? It's pretty complicated. Hopefully people can understand.
Well, I think they may be confused. So you're showing that the rise in current is instantaneous, but you could also say, so that would go away if you had the catalytically dead domain, and you would have no instantaneous
yeah,
rise.
This is the same. This is the current from this channel.
Okay. Right.
So from this channel. So when we take this actual current right here and we take the original current trace just to see based on time, this is based on pressure, but if we do it based on time, we can calculate when this first when we should first see current. And, you know, we're taking this, the initial rise in current and seeing when do we first see it. And there's an instrument delay here from when we first apply the pressure. And then when we have catalytically dead, there is no current. So,
I'm a little surprised that so people do a lot of like biophysics modeling of the membrane and how fluid it is and how quick it moves. I'm surprised people doubted that it could move that fast.
It's how close they are together.
Okay, it's about the proximity.
Yeah, so you have to understand they're very close together to begin with, but they're separate. So when people were thinking about 250 nanometers before, they were thinking 250 nanometers versus being somewhere else, very far away. And now we've gone down to a whole new world that is really close together. And I mean, if it's 10 times closer, the diffusion rate would be many-fold faster. So it just depends how close you can get without actually mixing. It's kind of like, for engineers, it's kind of like the problem of improving circuits by getting smaller and smaller. They go faster and faster, but at some point they're so close together that you lose the ability to distinguish whether you're on or off. Kind of like that.
Can you experimentally manipulate the distance by reducing, let's say, the number of these domain clusters, or have you?
We haven't done that yet, but yeah, if we change the amount of PIP2, we should be able to do that. It's a really good question.
Just like stretch it out a little. Okay, that's all my questions for now.
So you want to do this slide?
No, I'll let you do it. And also, I think Nick really developed. Oh, sorry, this is the arousal.
Yeah.
Okay. So let me say really quick.
This was Keith helped with this one a lot.
Yeah. So Scott and I are close because we worked together back in the day to develop paradigms for assessing these channels in vivo. So we use the fruit fly, which is an animal model that's really easy to knock genes out and manipulate them. And Scott had all this evidence in slice and in cell culture and whatnot. And that's amazing, but does it actually affect the way an animal behaves or senses force? And I had done a lot of work on fruit fly sleep, where we would deliver vibrations and ask, does the animal respond to that vibration, or is it asleep and not notice? And I think we met over like a drink, like Bill had us all at some bar.
Yeah.
And me and Scott just started talking and I'll let you go from there. No more history. But.
So he developed this system to apply more and more mechanical force. And so then the question is, when we knock out this enzyme, this PLD enzyme, is the animal more or less sensitive to force? And it turns out that it's more sensitive to force, which is what we'd expect. And you can see here that since it's more sensitive to force when it's knocked out, it responds to a lower threshold. And it was statistically significant. So even this one enzyme in the brain, and this is mechanical force, right? It's responding to mechanical force. When that enzyme that's needs to move out and set those thresholds using track, well, there actually isn't tracking the fly, but the PA is what's controlling that threshold. So anyway, I included that because it's an in vivo manifestation that these mechanical forces do actually change the behavior of an animal. Okay, so the last little bit that we'll talk about regarding track is membrane thickness. So if the channel moves from thick membranes to thin membranes, then that should affect this gating helix. And this can be substantially thicker. People don't think about this, but like 10 angstrom thicker. So the membrane is only 40 or 50 angstrom thicker. So it's a substantial portion of the membrane. It dramatically changes thickness in between different lipids. And so we first just tested to see whether track was in the GM1 domain. So I'd switch the colors on you here. The red is now the GM1 domain. but we wanted to say, oh, is track in a GM1 domain? It binds PLD. Why wouldn't it go to a GM1 domain with PLD? So we tested that. We labeled track and we labeled the GM1 domains with this color toxin. And surprisingly, we didn't see any interaction or any correlation. And we were surprised. But it occurred to us in our tissue culture dish, cholesterol is very low. Cells don't have a lot of ApoE, which is what's required, to load cholesterol into cells. And people that are working in cell cultures, I think eventually are going to appreciate how just deficient the amount of cholesterol is, especially when you're working on neurons. These are primary neurons from a mouse brain. And brains have really high cholesterol. It can be up to 45% of all the lipids in the brain is cholesterol. So that's a lot. And if you're not delivering the right amount, then these domains don't behave properly. Or if, say, you had an Alzheimer's disease patient that has high cholesterol in the brain, that is going to affect how all these domains work. And so we wanted to test this and see if TREC would go into the domain. So we took ApoE, which is a protein that delivers cholesterol. So we loaded the ApoE with cholesterol, and then we applied it to the nerves. And we could see that then TREC dramatically increased its association with the GM1 domain. So TREC is going into the domain when there's enough cholesterol. And you strengthen the affinity of PLD for that domain. And then we applied mechanical, so that's right here. Then we applied, this is just a summary, a percent summary of this, that it increases. Then with cholesterol, sorry, this purple should actually be green like that. So then when we apply this same shear plus cholesterol, the shear counteracts it. So the cholesterol is helping the ion channel go into the domain, and then the shear helps it come out. And we think this is what's going on with ultrasound, that these ion channels can go in and out of these domains. And then when you apply the ultrasound, it's causing the ion channels to shift out of those domains. What happened? Oh, this is the electrophysiology showing that when it shifts into that domain, it's inhibited. So the brain can use cholesterol to shut a channel off, and then mechanical force is used to then turn it back on by moving it back out. So that could either be, remember those forces we saw at the beginning where the brain is pulsating and there's all kinds of forces going on? Those forces are probably real, and they're probably part of this equation of things moving in and out. And then you apply ultrasound on top of that, and now you're going to manipulate how all these channels come out and what fires and what that's our model.
Scott, this is a less sciencey question, but when people are said to have high cholesterol, is that just in the blood, or is that also in their cell membrane? And would they be more or less affected by mechanical force?
Oh, that's a fascinating question.
Yeah, it's not super serious, but
this is a big problem. We've been talking to a pulmonologist, and we're looking to develop an assay to actually look at cholesterol in the tissue. I think of it like a harbor. You have ships coming into the harbor, but once you load your warehouse is loaded. That doesn't mean there's any ships in the harbor. So if you look at cholesterol in the blood, that's a problem. Actually, sometimes when the warehouse is the most full is when the harbor is the least busy. And we think that that's what's happening with cholesterol.
You're abstracting this so far that I'm starting to get confused.
No, the ships bring the cargo in, the cholesterol. Your blood delivers the cholesterol to the cells. And once the cells are full, we think that the blood cholesterol can go very low. And it's a false sense that the cells are empty.
So the cells are the warehouse then?
Yeah, your cells store the cholesterol. And we do think that that's what's going on in Alzheimer's disease. So, but to answer your question, the blood doesn't necessarily match the cell. But the other question is okay, if TREC now translocates over to PIP2, two things happen. It, of course, gets activated by PA, as we discussed, a lipid, but it also gets thinner. And so we checked this to see, well, could the thinning be helping activate the channel 2? And we purified the channel. We put it into liposomes of very defined thickness. The thinnest membranes were 16 and the thickest were 36 or 33 angstrom thick, this 22 and 16.1. And you can see that the channel, this is the data. I don't need to go over it for interest of time, but we're just measuring current through the channel in a flux assay. And then if we plot that flux, you can see that there's a very particular thickness where the channel opens the most, and that's in these relatively thin membranes. But if you get too thick or too thin, the channel inactivates.
Scott, I don't mean to keep interrupting, but that's super interesting. Does that suggest that applying too much mechanical force will not have an effect on this channel? And does that show up in data anywhere besides this, or?
Yeah, that was our original hypothesis. And it turned out that the stretch that you put on a membrane is very limited. So it can only change a few, like 1 angstrom, or at least that's what I'm told by the biophysicists. So you actually can't put that much, you can't change the thickness very much by stretch. And that's where we came out. So we'd actually done those experiments and it wasn't very compelling. And we submitted the paper for review. Somebody pointed it out and we're like, yeah, something's wrong here. And that's where we realized, oh, if it's translocating between thick and thin lipids, that could be a difference of 10 angstrom. So can you disrupt past disruption? No, it's probably already disrupted. So we wouldn't predict you could thin too much. And we haven't seen that in the data, but we actually thought the way you were thinking. And then when the data didn't support that, then we had to go back and be like, oh, maybe it can't thin that much. So it is that model is supported by data, but it's not exactly what you're thinking. Cholesterol is dynamic in cells. It goes up and down with each meal in the blood. And so we were interested in how cholesterol would change the thickness. And it turns out that if you add 30% cholesterol to the 20, it completely inhibits it. And remember the 16 that was the thinnest? Now when we add 30% cholesterol, it's fully open. So cholesterol shifts where the thickness of the membrane. And loading and unloading cholesterol is probably affecting many things that are transmembrane proteins. Okay, and this is just summarizing that. I don't think I want to go into too much detail over this thick and thin. The next 3 slides are just to kind of bring everything together and explain, when you do your experiments, how this thick and thin could be influencing your result in animal models. So we'll talk about dietary lipids that you might be feeding an animal. We'll talk about anesthetics that you might use on the brain of an animal, and then how those things work together. Now, so dietary lipids, if you feed an animal arachidonic acid, we looked just on cells and we could see that the arachidonic acid goes straight into the membranes. This is just using mass spec to see arachidonic acid going in. And this arachidonic acid activates TREC channels. That's what's shown here. So this is current TREC is getting activated. And we see when we did lipidomics on that, we can see the arachidonic acid goes into the PC lipids. So as you eat healthier foods like fish foods, your membranes get more and more of those type of lipids, and you change the chemistry because you change the balance between saturated lipids and cholesterol and polyunsaturated lipids. You can have diseases like ApoE and Alzheimer's disease that delivers the cholesterol and is changing the amount of cholesterol in the brain. And we think then you're moving the entire spectrum over to a saturated type. And if you eat the fish oils and stuff like that, you shift it over to this type of signaling, the PIP2 signaling. These concepts, so then ultrasound could play in this exact same way, that it's counteracting high cholesterol, basically. Mechanical force is counteracting this high cholesterol. That would be our hypothesis. This is just a model right now that based on these initial results out of how TREC channels work. I mentioned it, anesthetics. We did a whole nother study, and I have a reference at the end, you can read this, but we found that anesthetics also disrupt rafts. Remember, we think that this is a fundamental property of membranes. All nerves have this response and have these lipid domains. everywhere. So if the cell is using them to control ion channels, it wouldn't be surprising that there's other things that are interacting with these lipid domains. And this is the exact same experiments you've been looking at. This is PLD and GM1. And of course, in our controls, we see it highly correlated. And then we add chloroform and isoflurane, and they completely displace PLD out of the lipid raft. And this is highly significant. And we think it's a little different. Instead of disrupting it like with mechanical force, the anesthetic molecules actually go into the domains and displace the palmitate lipid interactions. We have a paper on that that's in PNAS that you can read about how those anesthetics would be interacting with the domains. But remember now, our prediction would be if you put these animals under anesthesia and you add ultrasound, you're going to reduce the that you need the ultrasound because this interaction is already disrupted by the anesthetic. We did the same experiment on trials where we added the catalytically dead PLD. So this is current chloroform. It's an anesthetic sensitive channel. It activated. And then when we added the catalytically dead PLD, it didn't respond to anesthetics anymore. So we think that anesthetic sensitivity is related to lipid raft disruption. And I think with that, well, there's lots of channel. I have 2 more slides. They're a little complicated, but I think it's valuable to put everything together. So ion channels, there's lots of ion channels that are palmitoylated. All you need is a palmitoylated protein that has both palmitylation and PIP2, and it should behave identical, right? If it's got palmitate and it binds PIP2, it should undergo this shift. And all these channels have that type of interaction. GABA, NMDA, they're palmitoylated and they have PIP2 binding sites. So we think that this needs to be studied in a broad spectrum. So how could this change how could this come to stay with me here? This is the last slide.
We're with you.
So this is how we think it works. You have an excitatory channel. When it comes out of the domain, it encounters a new lipid. For the potassium channels, it activates it. For the calcium channel, it inhibits it. And this causes anesthesia. But in another type of nerve, you could have a channel that's activated by this lipid. So now, remember, this is an excitatory pain channel. So now when you disrupt the lipid, all the channels are sitting in the GM1 domain in this model. And when they come out, they all bind to some lipid. And it's just whether the lipid activates them or it inhibits them. If you have a sodium, the neuron by disruption. If you have a potassium channel, you're going to inhibit the neuron by disruption. So of these 2 things, if you have a lot of potassium channels and this type of channel, it's going to be closed and you're going to have all potassium on the outside and you're going to get anesthesia. If you have this combination of channels, then you'll have very little potassium on the outside. And we think this is the setup of a pain channel. And what's the evidence of this? When you take an anesthetic like alcohol and you put it on a pain fiber, what happens? Keith, you ever got alcohol on a cut?
It enhances the pain sensitivity.
Why would an anesthetic cause pain?
I suppose if you have more excitatory channels, it's a cell type question, isn't it?
Yeah, you're right. You're exactly right. So in our model, again, this is just a model, but if both excitatory and inhibitory channels are sitting in these domains and you release them all with anesthetics, if you have more pain channels than you have potassium channels, you're going to get excitation. and you'll fire the pain nerve. It doesn't matter that it's an anesthetic. It's disrupting the domains and you fire the pain channel.
So you're saying, Scott, that all cell types are going to have some different response to anesthetics, depending on what they are.
Because they're going to have different combinations of these different channels. Now, there would be lots of other mechanisms at play too. We're only talking about a membrane-mediated mechanism, but we think that it could be important generally. And if those things bear out, this could explain a lot, or even all of anesthesia models, and they all come together. In lecture, we just wanted to talk about how could the membrane be the target? How could a membrane cause these different excitation and inhibition? And so with that, I can either wrap up. That's the material that I had to share. There's some suggested reading. The study on anesthesia. This is the first study showing by Nick, showing mechanical activation of an enzyme. And then if you wanted to dive deeper into understanding how those different channels could be expressed and how they could give rise to different activation, that's in this paper right here, disruption of polymer. And then these are just kind of background of how work, and this is a good review on just lipid membranes. So yeah, I completely lose you.
Kim, do you want to ask questions first, or did you want me to? I have a bunch of, well, at least a few questions, and anybody watching can check out or hang around for this portion.
I had a couple questions. I was just wondering.
Yeah, go ahead.
Yeah, I was just wondering about. Well, first of all, so you're talking about this effect when there's both the PLD enzyme, the PIP2, and the TREC. Do they all go together, and are there certain cells that have all of these, or do all cells, all neurons have these?
So all neurons have PLD. PLD is in every single cell. TREC is not in every single cell. We actually just recently did the experiment where we added cholesterol and we saw TREC go the GM1 domains. So really low cholesterol. There may be some, they may be in a complex, but if they are, it means TREC pulled the PLD out of the complex. And we know, or out of the GM1 domain. So the complex pulls PLD out of the GM1 domain, we think. Or they're separate. But when you have enough cholesterol and all of the PLD is in the GM1 domain, we think that there's enough PLD in there. There's more PLD than TREC, we think, naturally. And so then that kind of pulls TREC into the membrane. But yeah, we think that they move around as a complex now. We didn't think that originally because we didn't have the data showing that they went in together. And it means that PLD bound to TREC is less likely to be in a GM1 domain than PLD on its own. It's kind of it's been tricky to see exactly where everything is because there's 4 parts. There's PLD, TREC, 2 domains. And TREC and PLD aren't always together.
Okay. Yeah.
So this is a side note. I wanted to point out your line on the bottom. Go ahead, Kim. Sorry.
Oh, no, go ahead.
No, you keep going with questions.
Yeah, sorry, the connection's not all that great. So yeah, another question just to follow up on what you were saying is that about the amount of cholesterol affects the sensitivity to shear. And so what about like the effect of age and or exercise and sensitivity to mechanical effects?
We've done those studies in mice, and as you age, you load more cholesterol, at least in the lung. Well, we've done lung and liver now. So the blood cholesterol tends to go down a little bit with age, but the cellular cholesterol, the chemistry inside the cell, actually increases in cholesterol. Exercise, we haven't done yet. My just gut instinct based on the response of exercise to bad cholesterol situations is that you probably have more of these polyunsaturated lipids with cholesterol. So eating healthy and exercising, we expect to, certainly eating right will increase those polyunsaturated lipids in the membrane and shift it away from cholesterol. But exercise, since it does the same thing as eating right, and this has been shown over and over in multiple diseases, we think that exercise is going to do the same thing too, though. And we know the molecules and we want to study it. That's on our list.
Okay. All right, go ahead.
Yeah. Just for interest in COVID, COVID-19, the virus uses this same entry pathway. These GM1 domains are the one of the entry pathways for COVID-19. And so if you eat right, and presumably if you exercise, like you were saying, Kim, that that you move more towards the disordered region, the receptor for the virus moves out of the GM1 domains, and then it's harder for the virus to get in. So we think that this is why age and people who are obese are getting infected more. Is this?
Hopefully the NIH is watching. That's a pitch from Scott. You'd like more funding for this. Scott, I wanted to ask a couple questions. Do you think that circuits that are critical for survival, like brain stem circuits that you need for breathing during anesthesia, are they anesthesia resistant because they encode less of the and anesthetic that you're talking about?
This is a fantastic question. Let's think through this together. Okay.
I don't have anything. Yeah, I just want you to tell me.
No. You're gonna help me. You're gonna help me. They're easy questions. Okay. Let's think through this together. So if you want to survive, your brain can't run out of energy, right? It cannot run out of energy because once a nerve dies, it's dead, not coming back. So what would you protect against? Which would be come first? Would you run out of energy first, or would you go to sleep first?
You would go to sleep first.
You'd have to go to sleep first. There's no way the brain run out of energy before you would go to sleep. So the default mechanism has to be
higher energy using cells are more anesthetic sensitive.
Yeah.
Huh.
It would have to, right?
Yeah. Has it been tested?
It would evolve, it for sure would have evolved that way. Yeah. Huh. And that should bear out. So, and then again, this is a big if, but if ultrasound is working the same way as anesthetics, it would also mean that those regions where you apply ultrasound in the high energy regions should also be able to disrupt rafts more easily.
Super interesting. So another question, Kim and I have both probably seen a number of papers, I'm sure you have, where depending on whether or not the animal is anesthetized, the effect will go away, it may come back. And this could be, of course, because, the circuits just can't operate. But it also, could be entirely from what you're showing here. So my question is, and it's like I'm asking for experiments to do here, but what path would you pursue in terms of using anesthetic to sensitize the brain to ultrasound? I mean, there's so many interesting things from your lecture that I think we could pursue here, but.
So I think the first experiment that would need to be done would be with anesthetics, trach, and ultrasound. So that group at Stanford that has that ultrasound set up, we should definitely talk to them. And I had proposed that experiment in a grant a long time ago, and I never got the ultrasound set up. And this class has really motivated me to make that happen. So we probably will do that. And then I think once you, if we could definitely pin down, you'd want to do this with data first, show that anesthetics and ultrasound are working in the same pathway. Then you would want to start digging through the anesthetic literature and start looking at how, because there's been a lot of stuff done with anesthetic on the brain and how that's working, and that could guide you into always at the same. I should mention one thing though. anesthetics are only disrupting palmitate mediated localization. So it's only disrupting GM1 processing to our knowledge. Mechanical force, we haven't published this yet, but mechanical disrupts GM1, it disrupts PIP2 and PIP3 separation. So when I didn't show this data, but when we mix, or when we cause mechanical stimulation, PIP2 and PIP3 domains lose their separation. So if there's enzymes and proteins that are separated by PIP2 and PIP3, those would be affected by ultrasound, but not by anesthetics. And so you're going to get different, ultrasound is going to be different than anesthesia for sure, I think. Ultrasound is very interesting in that regard.
Okay, so if you look at the bottom here, Scott wrote, if nobody thinks any differently, is it really worth doing? And I wanted to point out that he's writing that because people have been fighting Scott tooth and nail for like a large portion of his scientific career. And what he's just shown everyone is fundamentally a paradigm shift, is the mechanical force sensor to the membrane, as he's saying. So, you know, take it as that you're learning really like what's happening right now, and sort of what may be really the future of the way we think. And my question around that little rant is, are people fighting you as hard as they were when I was over at Scripps and we used to talk about this?
You know, big claims require really big experiments. That, I mean, if you're going to make a big claim, you got to prove it. And you know, I do complain a little bit. And yes, yeah, people say things, and I look at it, I'm like, no, that does not. There's been nothing that I look at and I'm like, oh no, we can't explain this. Right? You look back at the ultrasound go back and look at that, the ultrasound on track, and you start thinking about it with this enzyme coming out, showing that it's fast enough. Like everything that people complained about in the beginning, you know, one thing after another falls right into place. And so, but it is important in science to, you know, everything's got to be backed up. And you don't, I feel comfortable with this now because we have at least 3 paradigms. We have anesthesia and we have mechanosensation. And so if you look at those 2 and we have good experiments in both of them, then it does look like that paradigm shift will come someday. But, you know, most people who do mechanosensation aren't looking at anesthesia, and they can't look at those other experiments and realize, oh, wow, this is working out this way in anesthesia. I would say, is it really worth doing? It doesn't mean that just because you don't change somebody's mind doesn't mean it's not worth doing, but it better be important for other reasons. And we try to do the experiments that will change somebody's mind, that somebody will think differently after. And I think those are the best experiments. When you walk away and you're like, wow, I think very differently, even if you're not fully convinced, even if your mind is saying there's a new world here to me and I hadn't considered it this way. Then when you go and look at your own experiments, those thoughts will creep back in and you'll be like, oh, wait a minute, maybe this is right. And then you can go in and do a new set of experiments. And I think that's why it's important to convince people to consider different ways of thinking, even, you know, up until they're fully convinced. It is a rough, it's a rough road to row, rough road to hoe. I grew up on a farm in Idaho, so a little bit of an Idaho reference there.
So, very nice. So yeah, I'm glad we had you on, and I hope a lot of people see this lecture.